Hi guys, and welcome to part 4 of vessels. We just finished naming some of the arteries, and what we're going to move on to now is capillaries. So capillaries are the site of exchange. So nothing else really exchanges materials or delivers oxygen at the site. Only the capillaries do that. The rest is just transport. Arteries and veins are getting it to the capillaries, and it's the capillaries that are going to allow the passage of gases and glucose and all that. So this is the most intricate network of vessels, right? Because this is where all the exchange is going to be happening. And we talked about them in the beginning when we talked about how thin do you want to make that layer. If you're going to have transport, you don't want it thick. You're going to want it very thin so that you can allow these molecules to be able to pass through easily. So there are essentially 3 types. We categorize them as 3 different types of capillaries. They can be continuous, they can be fenestrated, or they can be sinusoid or sinusoidal. So let's look at those individually. The most common capillary is the one that's called continuous, the one that has actually more limited exchange. And I want you to look at the wall of this. This is a cross section, correct? And how do you build one of these? It's just made with a wall made up of cells, correct? So you have here one cell with its mitochondria, and you notice this is one cell ends there, and there's another cell that ends there, and there's another one here, and you can see the nucleus. And this is another cell that's sort of aiding it. But think of the yellow here. All the wall of it is just made by 1 cell, 2, 3, all in single layer. So these are going to be able to allow the passage of substances going across them. Now look at the width of this. It's barely the width of an erythrocyte, of a red blood cell. So only 1 fits at a time. That's how tiny these are. Now we're going to want solutes to cross this membrane. So if I get my pen here, they're going to be able to, somehow exit here, and some materials are going to be able to enter here because this is so very thin. So this continuous type has an example. We have these in muscle, we have these in nervous tissue, and it's going to allow the passage of solutes. It's also going to allow the passage of glucose. Okay, so let's make a big G for glucose is actually entering the cell. There's various ways that it can do that. Like, do you notice where this is joining that one? So maybe it can scoot in between, right? Or maybe it can form a little vesicle like this and then just travel across and get inside. But somehow it's going to have to cross this barrier. Okay, continuous, think of it as the most common and the least exchange, even though a lot of exchange is happening. Okay, in a fenestrated capillary, it has something called fenestrations. So if you notice, it's kind of like extra pores that it would have here. So, and here also. Do you notice? So instead of being limited to the passage between where these 2 cells join, or across this membrane and into the inside, on top of that it has these other little openings that are going to allow for greater filtration. Where do you want those? Where is there a lot of filtration? There's a lot of filtration in the intestines. There's a lot of filtration in the kidneys, right? It's filtering all your blood, 160 liters of it every day. So you're going to want these to be a little leakier, and it's going to allow for that filtration and absorption. So this, think of fenestrated as having fenestrations and being found in the intestines and in the kidney. The third is sinusoidal. So these are very leaky. So what else do they have? Well, they have those fenestrations. They also have these pores, filtration pores. And then also it's going to allow it to cross the membrane itself. Do you notice it's doing like the other ones did, which is going through the cell and out the other end. And then it's also going to be able to go out where one cell meets, and here's another one where the cells don't exactly meet tightly. So there's that passageway. But remember that on top of that, there's these special pores. It's very leaky. Okay, these look very twisted and irregular, these capillaries. They would be in a place where you want to filter large things. And where is that? The liver, the spleen that's filtering blood all the time also, and bone marrow. Think of bone marrow. It's a place where you make red blood cells, and then that red blood cell has to scoot through. It has to leave that area in the capillary, so it's got to have a place where it can actually exit out, right? So that's going to happen in these sinusoidal, leaky capillaries. Proteins are large molecules, and they too can cross these membranes. So less common. A capillary bed, we refer to a bed as an area that has many of these capillaries, anywhere from 10 to 100 hundreds of them. And what you have is an arteriole, right, that's supplying a region here, a region of the body. It could be muscle or whatever. And it's going to supply, and then turn into venous blood and reach the venous system. So in that area that it's transforming from an artery to a vein is where the capillaries are leaking out and distributing oxygen. Now, I want to tell you what a precapillary sphincter is. What happens is that sometimes this region that I showed you here is not in need of oxygen or of nutrients, in which case we could just shunt that blood that's coming from the artery straight to the vein and just keep on going, right? And just bypass this, and maybe a little bit later open it up. But capillaries don't have the ability to vasoconstrict and vasodilate because they don't have muscle. So what happens is that this one center river here, which is called the meta-arteriole, this meta-arteriole does have muscle. So this is sphincter here, and it can close this off and not allow the distribution of oxygen in this area, just bypass and keep on going. So those precapillary sphincters are muscle that can close down and limit the volume of blood to a certain capillary bed. Why would you want to do that? Well, sometimes you are not digesting and you don't need all that oxygen to those organs, or you're shunting it over because you're running and you're shunting it over instead to your muscles. So you want to have the capacity of distributing blood where you want to distribute more of it. This is an enlargement of that capillary bed. So that meta-arteriole is that middle one with its little, ability here to close that off, because the muscle could squeeze it tight and then it's a sphincter. Or it can just go through all the channels and distribute to all the cells that are in this vicinity, right? Let's imagine there's lots of cells here, and these are all getting irrigated by this one particular capillary bed. And if there's another one over here that's sort of out of there, then you're going to need another capillary bed in that area. So every single cell of your body needs to be in close proximity to a capillary bed. Okay, let's stop there, and our next video would be on veins. Thanks for listening.